everyone. Today I am going to tell you about complement system from the chapter of immunology, one of the important topics. So I have divided it into multiple clips to make it easy. First of all, we will see the definition of it. Complement system is the system of the 20 amino acids and 20 proteins which are present in the animals or the human serum. And what is the ability? Its ability is to augment the effect of the components of the immune system, for example antibodies. Okay. Then we have the nature to discuss. The components of the complement system are such that whenever antigen comes in contact with the first component, it will activate the second one, second will activate the third, third will activate the fourth, and so on. So it will go in a cascade fashion. That is why it is known as the complement cascade. Okay. And components of the complement system are present in the form of the proenzymes, which are activated. First component is activated by the antigen, while the remaining are activated by the preceding one. Okay. Then we have the components of the complement system. We have the 9 components ranging from C1 to C9, named as C1, C2, C3, C4, C5, C6, 7, 8, and C9 at the end. And in C1 and every component, C stands for complement, not the component. Okay, C stands for complement. C1 has further 3 subunits named as C1Q, C1R, and C1S, where C1Q is specialized for the antigen binding. So antigen binds to C1Q and activates C1R. C1R activates C1S. Okay. And one of the thing that I want to tell you there is that C1, it is heat labile. It is heat labile. Okay. And it is present in the euglobulin fraction of the serum. Now, what is the euglobulin fraction of the serum? Euglobulin fraction of the serum means whenever we change the pH or we change the salt concentration inside the serum, then we will see the precipitates of some of the proteins, and those proteins are known as the euglobulin. Okay, we will see in the remaining topics, so don't worry about it. Then we have C2. It is also heat labile, but it is not present in the euglobulin fraction of the serum. Rather, it is present in alpha and gamma fraction of the serum. Then we have the C3. C3 is not heat labile. It is heat stable. C3, it is stable to heat, and it is not present in the alpha and gamma fraction of the serum. Rather, it is present in the euglobulin. Okay, euglobulin. And talking about C4, it is heat stable. C4, heat stable, but destroys, it is destroyed on let me tell you, it is destroyed when we have ammonia, NH3. Okay, NH3 ammonia, and hydrazine NH2. When we have blood with NH2 means hydrazine or NH3, then we will be deprived of C4 component. Then C4, C6, C7, C8 and C9. They all constitute MAC. Now what is MAC? MAC stands for membrane attack complex. We will discuss it in detail. And these are required for the final show, and the final show will be discussed in the remaining parts of the complement system. Okay. Then we have the complement number one. Okay, complement number one has three components: C1q, C1r, C1s. I have told you before that C1q is attached, or antigen is attached to the C1q and is activated, and in turn C1q activates C1r and C1r activates C1s. Okay, now the last slide of today. There are two pathways to activate the complement system and to provoke the response against the antigen. Number one pathway is the classical, and number two is the alternate pathway. So we will discuss these two pathways in upcoming lectures about the complement system. So stay tuned if you want to get some knowledge about the complement system. Thanks for watching, and don't forget to subscribe, don't forget to like, don't forget to share, and don't forget to comment. Thank you very much. We'll meet you soon in the next video about the classical pathway of the complement system. Thank you very much.